Hi, my name is Zac Burns, and I'm the chair of the Discovery Committee at the Myhre Syndrome Foundation. Today, I'd like to give you an update describing how the work is going as we progress toward developing new treatments for Myhre syndrome. We're going to give a broad overview of the process, celebrate the progress that we've made in the past, and reveal some of the work that we're executing in the coming year and beyond. Before I get into details, I want to establish some context. The first question is, what are we doing? What is the goal of this effort? In a sentence, the Discovery Committee is on a mission to make the best treatments available to the Myhre syndrome community. The next question is why? Why is this a priority for the foundation, and why are we dedicated to pursuing this? Again, in a sentence, the Myhre syndrome community has a high degree of unmet medical need. What does that mean when we say unmet need? This term refers simply to the gap between the quality of life experienced with current treatments and the quality of life we could experience if treatments were excellent. Now, the degree of unmet need may seem obvious to many of us living with the disease. Many people watching today are those with Myhre syndrome or those with loved ones who have Myhre syndrome. But I still think it is a good idea to talk about the current state of affairs and paint a broader picture than the perspective of a single experience. By laying this out, I hope to provide a reference point to contrast with our desired outcome and be able to articulate exactly what it is that we want in treatment and why the treatment options we are pursuing are the best options for our community as a whole. The fact is, our community is faced with many different symptoms, ranging from annoying to life-threatening. We have strokes, intellectual disability, heart defects, scarring leading to the trachea closing, pulmonary hypertension, which is very dangerous, constipation, stiffened joints, shortness of breath, anxiety and depression, autism, and cancer, to name a few. These symptoms can be debilitating on their own, but the burden of care of managing these symptoms adds yet another layer of difficulty for patients. Some take many pills with a constellation of side effects. Many treatments are expensive, a problem which is exacerbated by the loss of income due to the time spent managing the disease. Patients and caregivers are plagued by fear and anxiety about the future and how the disease may progress. Many experience frequent hospitalizations and trauma. Furthermore, the fibrosis in Myhre makes many treatments inaccessible. In some cases, the current treatment options are such a mismatch for Myhre syndrome that the treatments themselves cause lifelong problems, or the patient may even fail to survive the treatment. Today's treatment options were not explicitly designed for Myhre. People continue to live with the symptoms of the disease and sadly still have a low life expectancy. For some of us, that's not good enough. We want better lives for ourselves and for our loved ones. I don't want to downplay the fact that existing treatment is a miracle of science and human compassion. That is true. And that we want better and more targeted treatments is also true at the same time. So we've now gone over the what. We want to make the best treatments available. We've gone over the why. There is high unmet medical need. But how? That is what I'm going to break down over the remainder of the video. This question encompasses what we have done, what we're doing, and what we will do. To provide some structure and a sense of progression for how all the different initiatives fit together, I want to present this framework that I'm calling the 4 Ds of drugs. In this framework, our project progresses through 4 phases. The first phase is diligence. In this phase, many tasks must be completed before one can even start working on treatment. For example, you need to understand the disease mechanisms and how they manifest before you can figure out how to intervene in that process to treat it. This phase is primarily science, but we will also break it down further and you will see some other aspects here as well. I've written here that diligence took 5 years. That's not entirely accurate, as Myhre syndrome was first described in 1981, more than 40 years ago. But progress was slow and sporadic at first, and has been much more rapid and consistent lately. For the purposes of this timeline, I'm calling the beginning at the founding of the Myhre Syndrome Foundation in 2019. The second phase is discovery. This phase is essentially a review of the technology available, matching it to what we learned about the disease in the diligence step. In this phase, we are reading scientific papers, talking to experts, and forming and evaluating the hypotheses that we want to test. We narrow down the many options to a short list of those with the highest possible impact and probability of success. This phase consists of less science than the diligence phase and is predominantly characterized as research. This phase has been an area of focus for the Myhre Syndrome Foundation over the past year. But like the diligence phase, it also started earlier, but it's been more in focus over the last year or so. This brings us to development, which is where we are now. I consider this phase to be a shift from science and research to engineering. We're no longer asking as many questions. Instead, we're solving problems. People are in the lab, cranking the process to develop the technology specified in the discovery phase. If all goes well, the output of this phase isn't an idea, but an actual treatment that we think will work in humans ready to go. I wrote down about 3 years here, but the length may be different for each project. We have 4 very exciting projects in this phase that have either already started or are starting imminently. Some may take longer than 3 years, but some may finish sooner. I'll go over each of these 4 groundbreaking projects later today. The last phase is deployment. In this phase, we have a treatment from the development phase that we think should work in humans. But one can't just go selling it in the pharmacy of the grocery store. There's still much that has to happen to get it to the patients. We need to interact with regulatory bodies who ensure that the treatment is safe. We must hold clinical trials and enable a path for payment, whether by insurance or other means. In some sense, this phase represents a shift from engineering to bureaucracy. Next, I'll be drilling into each one of these phases to talk about what we have done, are doing, and plan to do. But before that, I need to give a caveat. This 4D framework is a cognitive tool to help us understand the process. The reality is much more messy. Projects do not follow a straight line where tasks flow neatly from one phase to the next. Instead, many things happen in parallel. There will be setbacks, the outcome is not guaranteed, and we might run out of money, or the treatment may not work, etc., etc. So with that out of the way, let's dig into each phase. The first phase is diligence. Like I said, the diligence phase is about everything that has to happen before work on treatment can even start. First, we need to understand the mechanisms of the disease. You can't fix that which you do not understand. For us, that starts with genetics. We know what specific mutations in the SMAD4 gene cause the disease. We know how those mutations cause the gain-of-function behavior in the SMAD4 protein. We know where the SMAD4 is expressed in the body. Of course, there will always be more questions to answer, but I'm putting a check mark next to genetics here because we know enough to move to the next phase for some kinds of treatments, especially gene editing. And we plan to answer additional questions as part of the process of developing treatment. For proteins, I'm putting down an in progress icon because although we know a lot about the SMAD4 protein, including how the mutated SMAD4 protein interacts with the other SMADs, there are still some questions we would like answers to that will expand the available treatments worth testing. For example, does the mutation cause the SMAD4 protein to interact with any other proteins that SMAD4 doesn't typically interact with? To answer that question, we're in the late planning stages for a project with Shule Mo at Emory University that would map out how the mutated SMAD4 protein interacts with other proteins in the body. If that project is successful, it would give us a list of other proteins that we can target with pills, because there are already lists of pills that interact with the other proteins. We could then take the small molecules in these pills and test them on Myhre cells and see what happens. So understanding proteins is in progress. We know a lot, enough to test certain treatments, and we are currently working on knowing more to expand our horizons. Another thing that we would like to understand to help us develop and test treatments is how the cells function. There are few publications on Myhre cell function, and they are very limited. Nobody has ever taken, say, brain cells from a Myhre syndrome patient to measure how those cells behave and how that is different from normal brain cells. So that's what we're going to do. We have yet to start this project, but we have started on the projects that feed into this. Earlier this year, we finished developing iPSCs, which are patient cells that can be instructed to grow into other kinds of patient cells, including brain cells. We finished these, and we've just started on the work to take the iPSCs and change them into other cells that we need. Once that work finishes, which should be sometime early this year, we will be able to start a project that measures cell function. Another thing that needs to happen is to establish a foundation. This work might seem boring, but it's critical. One needs to lay the legal and process groundwork to establish an entity that conduct the business of pushing forward treatment. You need to establish a company, registered as a nonprofit, set up email addresses, hire an executive director, recruit a volunteer board, develop a website, and so on. Without these, you'll be ineffective, and people won't take you seriously. MSF was founded in 2019 and has grown to be a competent organization, so that box is checked off. The next box here is fundraising. At MSF, our board is made up of volunteers. The only paid staff are our executive director and chief scientific officer. Because of this, we operate very leanly and cost effectively, which lets us set aside a large portion of our budget for research. If you look at the expenses for MSF in the 2023 annual report, excluding grant funding to advance Myhre research, we spent less than $36,000 and did quite a lot on that shoestring budget. But advancing treatment is different. The labs capable of creating the best treatments are not staffed by volunteers. They also require lab space and expensive equipment and materials to manufacture treatment. The budget for these projects dwarf the operating budget for MSF. We're talking about a jump from tens of thousands of dollars to millions of dollars. New treatments will cost us on the order of 100 times as much money than everything else that the Myhre Syndrome Foundation does combined. Yet there is a checkbox next to fundraising here. Does that mean we have all the millions that we need to complete the project in the bank? Unfortunately, no. Like many other checkboxes here, it means that we have enough funds to continue to the next phase. Specifically, we have enough runway to cover the commitments we have made with external partners in the short term. We wouldn't want to be unable to pay for a project that we are contractually obligated to pay, but over the next 3 years, we need to fundraise for the later stages of projects, or else the treatment they develop may be stalled in the pipeline. And there are other projects we would love to start, but we do not have the funds for them now. We can get into more details of the budget in the future update, but I want to clarify that the check mark next to fundraising means that we have enough to start on the next phase in earnest, not that we're remotely done with fundraising or have enough to finish the project. Next, we need to understand the manifestations of the disease and its progression. By this I mean, what are the symptoms and how do they develop over time? The natural history study is especially important for any sort of preventative treatment. Without a large patient population to use as a control in a clinical trial, we need to be able to see whether the likelihood of symptoms developing is reduced by comparing the treated population with some other group, like the group classified in a natural history study. For this, we have at least 3 natural history studies published. The largest followed 47 patients at Massachusetts General Hospital over many years, so we can check this box, mostly. There's an errata here in that it would be very beneficial to have a natural history study specifically for intellectual developmental delay and autism. The current literature on this acknowledges the existence of IDD and autism, but despite this area being a priority for many patients, it doesn't do as much as we would like to characterize the specific behaviors or nature of cognitive impairments that may come with Myhre syndrome. What we've done so far is to establish a working group and talk to caregivers of patients who are affected by IDD and autism to get an initial idea of how to design the study. And after their feedback, we found several PROMISE studies that we think could help track cognitive and behavioral issues often present in Myhre. PROMISE surveys are questionnaires that have already been approved by the FDA for use in establishing endpoints in a clinical trial. We're designing the NHS in a forward-looking manner to set us up for success in clinical trial. But we still need to finish the design of the study. We need to find funding, recruit patients, work with a clinician principal investigator, and finally conduct the study. Hopefully, if we keep pushing on this, we can have the results queued up before the treatments are developed. And beyond the IDD and autism observational study, there are other studies that we would like to do to enable treatment. The thing about the natural history studies is that they're very general, so they don't necessarily get into the detail necessary to run a clinical trial. They may acknowledge, for example, that stenosis exists, but not answer some questions that lead to endpoints in a clinical trial, like how much stenosis and so on. So we would like to do more observational studies, but haven't started on this yet. We would also like to conduct studies that would establish a link between biomarkers like skin thickness to other more severe manifestations of the disease like cardiac and pulmonary function. If we can do that, it will give us something easy to measure short-term improvements during a clinical trial that would indicate that a treatment likely addresses long-term problems. This is important to keep our clinical trial design within a reasonable timeline and budget. Yet another thing that we've accomplished in 2024 was to establish a biorepository. A biorepository is a facility that stores biological samples for research, like a patient's blood or tissue samples. For this, we are working with Combined Brain, who not only helps us with the logistics of collecting and storing samples, but they also bring an IRB, or Institutional Review Board, to ensure that everything we do is ethical, protects patient privacy, and complies with regulatory requirements. We also need a way to test treatments. Putting an untested treatment into a human is a bad idea for many reasons. So we need other ways to check if a treatment works and is safe. One way we test is with animal models. There are 4 animal models for Myhre syndrome. We have 3 mouse models and 1 zebrafish model, though the zebrafish model has yet to be characterized. So that gets a checkbox in that we have enough to get started. But we will soon start on a project to add a mouse model for the 496 variant. Testing on animals is a good idea, but animals are not humans. So we also want to test on patient cells. We can't test in a human, but we can test on their cells. The patient cells tell us how the cell is behaving in different parts of the body and how that differs between people with and without the disease. When we treat the cells, we can also check if the cell behavior improves. This is marked as in progress. What we've done over the last few months is to generate patient-derived iPSCs. These are cells that are taken from a patient's blood that we can turn into other kinds of cells, like heart cells and brain cells. This is better than trying to take samples from a patient's heart, brain, and other parts of the body. If you take the cells out of their vital organs, that's no good for patients. Instead, the patient donates their blood, and scientists turn those cells into other kinds of cells. Sometimes I barely believe the words coming out of my mouth. This is alchemy, but we did it in partnership with Combined Brain, who took the samples, and IXLs, a contract research organization who developed the iPSCs from the samples. The iPSCs are themselves a kind of cell model, so this has already enabled us to get started on treatment. But we also want to generate other kinds of cells, like heart and brain cells. So we just started a project with another contract research organization called NewSite, who will generate those cells from the iPSCs for us. It's a part of a larger project we call our preclinical platform, which is an all-in-one setup to let us test a variety of treatments in a standardized way. We expect to have these cells finished by early this year, which will hopefully be just in time for some of the treatments we want to test. Next in this category is assays, which is a fancy word for tests. For example, once we have the cell models, we need to know what we want to measure and how to measure it to check if the cells are behaving properly. One assay we might use is something called a scratch test. This test allows us to see how the cells heal. Do they form a normal scar, or does fibrosis occur, which would indicate Myhre? We won't know what the best assays are until we see the cell models. That's why this is listed as an item scheduled for the future. It is something we're thinking about now, though. Another kind of assay or test is not on the cell models, but on the animal models. For example, how do mice with Myhre behave? We've started to talk to some research organizations that work in this area, like transformation and psychogenics, to get an idea of the project. And we're looking for funding for animal model assays as well. Another way that we test treatment is to look for indications of Myhre in a patient's blood. These are called liquid biomarkers. For example, when someone has diabetes, you will find elevated levels of sugar in their blood. If the sugar levels in the blood are normal, it indicates that the diabetes treatment is working. Nobody knows what the equivalent indicators are for Myhre, so we're going to find out. The first step is to get enough samples of patient blood to have certainty that what you are seeing is statistically significant. The second is to analyze all those samples. We're still collecting samples. This is why at the conference we had Combined Brain and informed everyone of the opportunity to donate. We got a lot of samples, but we still need more. If you would like to help with this, please reach out. Depending on where you live, we may be able to get Combined Brain to send a phlebotomist to your home. We also recently started an initiative to work with people on the ground in Europe to get additional blood samples. Once we get enough, we can look into analyzing them. We've already started to plan for how we might analyze them, but this is still in the early stages. In order to start work on treatment, we also need to build a community. We need a network of patients, clinicians, scientists, donors, and volunteers that trust each other and work together. And we needed not just to connect these groups, but to connect them to each other, like connecting scientists with clinicians. This comes from a lot of hard work over time, connecting people and hosting events like the in-person conference that we had last year. Without this network, it's very difficult to get anything done. As you can see, the diligence phase is not done, but is ongoing. Work will continue even on items with a check mark. For example, fundraising enough to start the next phase doesn't mean enough funding to get to clinical trials. But overall, with a review of the progress so far, we can say that we're well positioned to focus on the next phase. We're pipelining the work so that parts that we haven't finished yet can be ready just in time while we start on the next phases. Now we move on to the discovery phase. Remember, the previous phase was about things that need to happen before you can start on treatment. This phase is research and reading literature and matching what you learned about the disease from the diligence phase to the treatment and technologies that exist and turning that into a concrete plan that leverages what you have built. The first thing that we needed to do was to hire a chief scientific officer with drug discovery and rare disease experience. Thankfully, we found Armel, who has been outlining and pushing forward a comprehensive strategic plan to obtain treatment. Working with Armel over the last year has been a pleasure. She is not only smart and experienced, but is also kind, empathetic, and caring for the rare disease community, and she knows how to get things done. It is easily stated that we would not have made nearly as much progress over the past year without her. With Armel's help, we also established a scientific advisory board. This board is made up of 6 experts in the field, like chemistry, gene therapy, stem cell biology, protein-protein interaction, and assays to gene therapy and more. We've been meeting with them monthly to discuss and refine our strategy, as well as doing grant reviews and answering more tactical questions to ensure that our research money is well spent. They have been doing volunteer work for us, and we are very grateful for their time and expertise. We've also done a thorough review of the scientific and regulatory literature about Myhre, SMAD4, treatment options, FDA guidelines, and more. A lot of time is spent just learning. One of the things I did personally was to spend a few days at the Ultragenics Bootcamp, which is an intense session where Ultragenics dumps information on how to navigate the drug discovery process. We're always reading papers, going to FDA informational sessions, attending conferences, and the like. This helps to make sure that we can go in the right direction and not miss any opportunities or spend resources on a treatment that won't be the best option. Another thing that we did this past year was to submit a request for proposals to leading institutions around the world in gene and cell therapy. We sent out the call and asked them, if we give you $450,000 over 3 years, what can you do for us? We gave some guidance about the kinds of therapies we were interested in, held meetings with everyone who was interested in participating, and got several very competent proposals back. These proposals were reviewed by our scientific advisory board, and we're deciding to move forward with one, which brings us to the next phase. That brings us to my favorite phase, development. Remember that the previous phase was research and planning. This phase is more like engineering. Labs are turning the crank to turn ideas and plans into actual treatments. Very exciting. There are 4 projects we're involved in that I'm going to share today. The first project is a cutting-edge technique coming out of Nationwide Children's Hospital called Remedy. Remedy uses your body's built-in DNA repair mechanisms to copy the patient's good copy of SMAD4 over the patient's mutated copy of SMAD4, using the healing mechanisms that your body already has and the genes that your body already has. There are several reasons why I think this is a very promising direction. The first is that it fixes the root cause of Myhre syndrome. In my experience as a system engineer, I've come to appreciate the value of fixing the problem at the source. For example, if you have a leaky roof and place a bucket to collect the water, you're going to have problems that you wouldn't otherwise have if you just patched the roof. You'll have to replace the bucket when it's full. You will have to navigate around the bucket as you traverse the house. Water will splash on the floor when it drips into the bucket, resulting in water damage, and so on. In my experience, I found this principle of the value of fixing the root cause to be broadly true across multiple domains, including medicine. If it does what we hope, it could be the treatment with the least problems. I want to emphasize again that we don't know yet, we have to try it, but this is part of my rationale for justifying trying this. Other factors where DNA repair holds promise include that it could be a proactive solution, not a reactive one. I don't want to wait for potentially fatal issues to get out of hand and then go to the emergency room for a heroic save. That's traumatic. I want to prevent problems before they happen. And as a father of a Myhre syndrome patient, I want as few treatments as possible. Every single treatment is another anxious trip to the hospital, another expense, and another needle in my son's arm. But DNA repair could treat many symptoms of Myhre in one go and benefit the most people in our population because all of the problems and symptoms are downstream of the root cause. In addition to the burden of administering many different treatments, we simply don't have the resources to create a different treatment for every symptom. Again, the promise of Remedy is speculative, but I think it's a reasonable hypothesis worth testing. And that's exactly what we're doing. We've already started a project with Nationwide to develop the treatment. If this continues to go according to plan, we will have the world's first corrected Myhre patient cells early this year. Once we get the corrected cells back, there is still much work to do, like seeing what percentage of the cells were corrected, whether the corrected cells function properly, trying the treatment in animals, verifying the treatment is safe, and getting through the whole deployment phase that I will talk about after discovery. But this feels to me like the home stretch in many ways, so I hope this works. I also hope that we get the funding required to bring us all the way to clinical trials. We think that testing for safety alone is going to cost us about $1.2 million. Those funds are not in the bank yet. Even so, I think that we're already trying to apply Remedy to Myhre is a very exciting development. The next project that we're working on is a cell therapy led by Genathon in collaboration with Imagine Institute. The idea here is to have a treatment that specifically targets fibrosis. When I say cell therapy, I mean that we would train the patient's immune system to attack fibrosis, not stem cells. We want to target fibrosis because it is a leading factor in the mortality of Myhre patients. Fibrosis is unnatural scarring, which doesn't sound too bad, but it is actually one reason behind stenosis and one of the reasons that surgery for Myhre patients is dangerous. If we can have a treatment that not only prevents fibrosis, but also restores tissue with excess scarring, then maybe we can help patients who already have fibrosis or who are at immediate risk. Another thing that makes this project exciting is that it is not using cutting-edge technology. It is using established technology. Cutting-edge technology can be great, but it also carries increased risk of project failure. Using something established means there will be more expertise that you can draw on, fewer unknowns, potentially a smoother path for regulatory approval, and so on. Another promising direction for us is pills. We've been working with an academic institution who already screened many thousands of small molecules against Myhre proteins to see which ones had an effect. That narrowed down the list, and the good news is that we have about 100 small molecules that we know will do something. The next step for these is to put them into our preclinical platform to test them against cells and see if they improve cell function. Every small molecule that we want to test costs money, so it's good to have a list about that size of 100 that's not so broad that it's prohibitively expensive and not so small that we're unlikely to find something. So there's been a lot of progress in this area of small molecules, and I'm hoping and looking forward to see what comes out of that. Our next steps are to finish the cells and test them in the preclinical platform and to get a grant to cover the costs of testing them. The last project that I want to highlight today is peptides. A biotech reached out to us saying that they want to develop a peptide treatment for Myhre and that they needed iPSCs to continue the work. They've been quite secretive about the results so far, but that's okay because it was very inexpensive and easy for us to support them by providing cells. This goes to show that we're in a position now where we can leverage our resources, like iPSCs, to support academics and to support pharmaceuticals. As a foundation, we do not have to be directly involved in every project and fund everything ourselves, yet we can still accelerate progress. Finally, the last section is deployment. This is mostly in the future, and I've already gone over a ton of information. So I'm just going to be brief right now and hopefully give another update next year with something more substantial. Remember, this phase represents the shift from engineering to bureaucracy. There is still a lot of science going on. Clinical trials, for example, are definitely science, but it's heavily regulated science. Once you have a treatment developed that you think works, one of the big questions is whether it is safe for humans. The study to check for this is called toxicology. You check how toxic the treatment is. Every treatment has some toxicity. Even an apple has some toxicity. If you eat apple seeds, they contain a chemical which produces cyanide. If you eat a lot of apple seeds, you will experience this toxicity. What you need to understand for treatment is how toxic it is, what are its side effects, and the crucial question here is, does the benefit of taking the treatment outweigh the risks? Toxicology is about answering that question without putting a human at risk, and it's expensive. This is where we think that we'll need about $1.2 million to continue with Remedy if it is demonstrated to work. Another thing that we're thinking about for deployment is establishing something called the freedom to operate. That is a lawyer term for making sure that we can continue to make steps toward getting treatments to patients legally. Specifically, this has to do with patent protections and whether the people we work with can tell us we aren't allowed to use the treatment. The sad truth about today's treatment landscape is that it's often not the science that gets in the way between people and treatment. It's bureaucracy and monetary interests. It's unfortunately quite common that a treatment exists but sits on a shelf somewhere where patients can't access it because one party with the patent rights doesn't want to go forward for whatever reason, which can include that it's just not their highest priority right now. And the patient advocacy organization, who just spent years and significant resources toward advancing the treatment, legally can't do anything but start over in a multi-year process to develop treatment, but this time with one less option. This is a terrible outcome, but a real risk that we have to factor into our operations today. So we've contracted with IP attorneys to help us navigate those issues and are putting the freedom to operate as a high priority in all of our legal negotiations whenever we can. This unfortunately slows things down now, but hopefully will de-risk the project later. Then, of course, there are clinical trials. These are a big task in the future and a very expensive one. This is something that we have barely started on. Even though it may seem far in the future, we are designing clinical trials right now because it is going to be a slow process and that we wanted to get started on this early. One thing we did within the last year is to host a patient listening session with the FDA. The patient listening session is a chance for patients, caregivers, and advocates to express their preferences directly to the FDA so they can take it into account when reviewing clinical trials. Patients express their current experience with Myhre syndrome and treatments, as well as their preferences for potential future treatments and clinical trials. Our listening session was attended by staff from 13 different offices from across the 4 FDA centers. They were very attentive, and I think that the work that we did there had an impact and will come into play when we're ready for clinical trials. Another thing that we're looking into right now is what kinds of grants can support our research, especially grants for preparing for clinical trials or even perhaps running the trial. One grant we're applying for is the DISC-0 Foundation Award, which is a program from CIRM in California. These are substantial grants, so we've been planning to position ourselves and our research in a way that we think can increase the likelihood of receiving a grant to fund later stages of research. We've also recently established a clinical trial working group that includes many clinicians who have each seen at least 4 Myhre patients. They've been working together to design our clinical trial, answering questions like what endpoints should we select, what observational data do we need, should we hold a multi-site trial, what kind of clinical trial do we want to run, and so on. Another initiative that we're working on is something called disease clustering. The problem that disease clustering solves is that it's difficult to run a proper clinical trial with very few patients. But with disease clustering, we can identify other diseases and other patient groups that have enough similarity to work with us together on a larger clinical trial. This is something that Armel has been working on as a part of a larger group, and the initial results are quite promising because it indicates we may find ourselves with more friends in the disease space and be able to work together. There's a lot more that we will need to do in the future here, but I think this is enough to cover for one video. I know that we have covered a lot and that there's just so much going on, but I think it's worth explaining why each project is important and how it fits into our cohesive plan to develop treatments and make them available. Lastly, I'd like to talk about some ways that you might be able to help. One of them is that you can donate blood for a viral repository. This is something that we've organized through Combined Brain, and though we have many samples so far, we don't have all of the samples that we need to work on liquid biomarkers. So if you're able to help with this, please reach out and I will send information about how to coordinate with Combined Brain. Another thing that you can do is participate in our registry with CORDS. That is an online survey that you can fill out yearly that helps us understand what symptoms are common and how the disease progresses over time. Another thing that you can do is to donate money. Even if you don't donate a lot, like I know I mentioned some big figures like $1.2 million for checking for toxicology, everything that is donated helps. Something like even getting a blood sample like I just talked about, we have to spend money to send out a phlebotomist to collect the sample, to ship it, and to store it in the biorepository. That doesn't cost a lot though, and your donation could enable that collection. Another thing that you can do is to participate in research, in working groups. I mentioned before the observational study, such as the IDD and autism study, as well as some other studies that we want to do in the future. We need participants who are patients or caregivers to participate in those studies if they're going to be a success. To be informed of those opportunities as well as other opportunities in the future, you can sign up for our newsletter on myhresyndrome.org. And lastly, if you feel that you'd like to help out with any of these things, or you have other ideas for how you might help, please reach out to me. My email address is zburns, ZBURNS, @myhresyndrome.org. Thanks for listening, and I hope we have another great year progressing toward treatment.